And this is now going to be a series of short 10-minute interventions from people from around the world. And what we will do is give 1 or 2 questions after each intervention, 1 or 2 questions of clarification directly related to the presentation. And then we will, after the last presentation, we have 6, then we will go into a more general discussion. So the first we have on the program is Martin Lema. Martin, are you with us? Maybe the tech.
Hi, Peter. Yes, I am.
Great. Good to see you as always, Martin. And just by a quick way of introduction, Martin, and you can correct me if I'm wrong. You're now in the academic world, but you were for a good period during the development of the Argentinian current policy on genome editing. You were working in the administration, if I've got this correctly. So that's your background. And thank you so much for making the presentation today.
Okay, so thank you, Peter. Hello, Erica. Hello, everyone. Yes, Peter, I can confirm that I was chairing the Argentine Biosafety Commission on the time that we developed our policy for genome-edited organisms for agriculture, which I'm about to describe now.
Thank you.
Mr. Lema, you can take the control of your slides by
clicking.
Thank
you. Okay, first of all, in order to develop our policy for new breeding techniques, including genome editing in agriculture, we started from recognizing that we...
Mr. Lema, I'm sorry, you did not take the control of the slides. Please click in the... Okay, please. Now it's working. Yeah.
Thank you. So as I was saying, we, Argentina, as most other countries in the world, have 2 established regulatory options for new genetic variants. On one side, for new varieties or breeds, including those obtained by mutagenesis, different kinds of mutagenesis. We have 1 regulator and 1 set of rules, and then we have another regulator and another set of rules for what we, for now, I will call transgenic organisms. We recognize that sometimes very similar products can be obtained by one or the other route. For instance, sorry, I'm trying to change the slide here. Here we have an example of a drought-tolerant crop obtained by conventional or traditional mutagenesis, if you want to call it that way, used in a different country, in Zambia, or we can have also a drought-resistant wheat, in this case developed by an Argentine biotech company and approved for commercialization in Argentina. So we started by recognizing that those 2 regulations exist, but now the problem with genome editing is that it created a question if the products belong to one box or the other. So as a brief introduction, our regulatory framework for modern biotechnology, let's call it for now, has been working for 3 decades now. We have over 60 genetically modified organisms, crops and recombinant live vaccines approved for full use in Argentine agriculture. And our regulatory commission is recognized worldwide as a reference in GMO biosafety. Particularly, the criteria for gene editing in agriculture that I'm about to describe is a case-by-case sorting mechanism to decide if each product is a GMO or not. It's based on the Cartagena Protocol definition for LMOs, or living modified organisms. This is the most worldwide recognized definition in this area, and it's the same as the Argentine definition for genetically modified organisms. The decision regarding if it is a GMO or not is made in 60 days. And if the product is a GMO, it has to be regulated as a GMO. But if not, it is under the regulatory framework for conventional products. As I was saying, the LMO definition from the Cartagena Protocol is a cornerstone to our approach. According to it, a genetically modified organism is that possesses a novel combination of genetic material obtained to the use of recombinant DNA techniques or direct injection of nucleic acids into cells, overcoming natural barriers. So we use this definition to discriminate line by line, product by product, if we are facing a GMO or not. The main question here in this very simple decision tree is there is a new combination of genetic material. If there is no new combination of genetic material, the product is not a GMO. So the applicant is notified about it, and then he must deal with the regulator for conventional products. Using this decision tree, we have been able to take already some decisions regarding several products from new breeding techniques, including genome editing in particular. In this map, you can see two dimensions. One of them relates to the concept of modern biotechnology in the Cartagena Protocol definition, and that's a vertical dimension. And then the horizontal dimension relates specifically to the concept of novel combination of genetic material. So this map allows us to classify the most likely classification that products will have, although the classification is made case by case by analyzing what is exactly the change made in the genome. In here, you can compare the dynamics of regulatory decisions, beginning in 1996 with the first approval of a genetically modified live product for agriculture in Argentina. And then since 2016, the decisions regarding if certain products from new breeding techniques, including genome editing, are GMOs or not. So blue versus red, you can see that products from new breeding techniques, which is mostly genome editing, have been presented to the regulators with a dynamic that goes much faster than GMOs. And also you can spot there those stars that highlight the fact that there has been some products developed by genome editing or other new breeding technique that when presented to the regulators in Argentina, it was decided that those products are GMOs. This is another interesting feature of the caseload that we have until now. As I was saying, most of what we call new breeding techniques, the concept is not specific to Argentina. It has also been used in the European Union preliminary studies about how to regulate these technologies. I was saying, among the so-called new breeding techniques, most of the cases that we are receiving are genome-edited organisms. And these organisms, if classified according to the biological kingdom and the trait of the modification, we can see that there is a lot of diversity. I mean, in the three biological kingdoms, and many traits, not only for agricultural purposes, but also for consumer preferences, increased health, also increase animal welfare, many diverse traits of interest to different sectors of society. If we compare that for a moment with the caseload of genetically modified organisms that have been approved over a very long extended period, 30 years, we can quickly see a difference in the GMO universe over 3 decades. Mostly we have plants in commercial use and some vaccines. And for those plants, the prevalent traits are only 2 of interest to the farmers: herbicide tolerance and insect protection. Only now we have some other traits arising the horizon, like drought resistance that I mentioned earlier. But then if we compare to new breeding techniques, genome editing, we can see that there is much more diversity in what can be offered to society in a relatively short term. If we now look at the profile of the applicants in the left, you can see that in the last 5 years of taking decisions regarding products from new breeding techniques and genome editing, most of the applicants or developers are local Argentine companies and Argentine public researchers. I mean, research institutions. That's very interesting, because if we compare that to GMOs, that's a minority in the GMO universe, as it happens in most other countries around the world. And then the other half of presentations received for genome edited products, most of course, all the other half are foreign, but most of that part is coming from small companies, not from those multinationals that have been mentioned before. But this doesn't mean that multinationals are not interested. They are also taking stage, but for some reason, perhaps they are not so fast in adapting or making use of new opportunities. We are seeing that most of the applicants and developers are from small companies or public research sector. In Argentina, as I was saying, we are seeing how Argentine scientists are developing products using CRISPR and other genome editing tools. Also, startup companies that were created solely to develop genome edited organisms for agriculture, or established companies, important seed companies in Argentina that now have created a biotechnology lab to have their own biotech crops using genome editing. And this is not restricted to plants. The same thing is happening in animals. Both the public sector and the Argentine biotech companies that, for instance, work on assisted reproduction and cloning now also have projects using genome editing. And this is because we are having this policy not only in Argentina, but in neighbor countries, as I'm going to describe next. In Argentina, we also, or the government also have work on the aspect of public communication. So this is made not at the back of the public, but public is participating and knows about these new policies. And I'm about to finish now. Argentina also is working with other countries, trying to find ways to harmonize and have the more rational approach for the regulation on genome edited crops and animals for food and agriculture. Finally, the same thing that I have described for Argentina is happening in other Latin American countries. In only 5 years, we have 8 countries in our region that have a very similar approach to the regulation of genome editing for agriculture. And of those, 3 of them, Chile, Brazil, and Colombia, are already receiving applications. And alongside with the governmental decisions, we can see that their public research and private biotech national sector are working on developing products of local interest. So with this, I finish. Thank you Reimagine Europa and all participants for the opportunity to share this with you.
Mr. Kearns, you have
Thanks very much, Martin. I think we have time for one question of clarification. And I think I have Rennie Custers. Are you with us, Rennie? This may take a second.
Please, Rennie, can you open the microphone and webcam?
Yeah. I'm here. Let me now properly introduce myself. I'm René Custers. I work for VIB, which is the Flanders Institute for Biotechnology. I indeed have a short question for clarification. In one of your first slides, you also mentioned you have the 2 systems. Either it is the conventional route that the product is taking, or the GMO route. But you mentioned that also in the conventional route, in certain specific cases, there may also be an additional risk assessment being triggered. Could you elaborate on how that is actually working?
Yes, for instance, in Argentina we have weeds that are from Brassica family, and when we have a plant having herbicide tolerant trait, that trait could be transferred to that weeds, and that may happen in both cases, if it is a GMO or if it is a mutant having a mutation that renders the plant with an allele with herbicide tolerance. So when the regulator of conventional new varieties obtained by any means of mutation, so maclonal variation, chemical, radiological, or even spontaneous, there is an application to register a Brassica crop having herbicide tolerance. That regulator develops the risk hypothesis that the allele can be transferred to the wild relatives that are weeds, and then that risk hypothesis is evaluated. And we have, for instance, a case where somebody tried to register sorghum having high cyanide levels to clear the fields from worms. And that was a spontaneous mutation, not something developed by using biotechnology. But that high levels of cyanide, aside from cleaning the field, also were a risk to animals entering the field. So there was a risk hypothesis for that. And we can continue. Microorganisms have their own route that always have a safety. I mean, microorganisms for agriculture, non-GMO, they have their own route for safety assessment, and animals can also have an activated system for risk analysis if the trait is so new that there is a risk hypothesis. So we have the same thing. If a genome edited product is decided that it's not a GMO, then anyway it has to be presented to the regulator of conventional products, and that regulator can activate a risk analysis, but only if there is an actual risk hypothesis. Moreover, our regulator, our GMO regulators, even when they may say this product is not a GMO, no more business in here, if they happen to develop a risk hypothesis because of the trait, they have the obligation to communicate the risk hypothesis both to the developer and to the regulator of conventional products for they to follow up on that.
Okay, thank you very much.
Thanks, Rennie, for the question. Thanks very much, Martin. Martin, stick with us, but we're going to move right along. And next it will be Dr. Vibha. Thank you for joining us today from New Delhi. I should mention at this point that some of our presenters have given us a lot more material than they will have time to go through in minutes available, but we will be making that available afterwards. Dr. Vibha, just by way of introduction, perhaps, and you can tell me if I am wrong, perhaps one of the most important points for you today is that, in my introduction, is that you are currently working with a government committee on developing policies and so on for genome editing. So if I'm not wrong, if I am, correct me, but please, as soon as you're ready, if we go with your presentation. Thank you.
Thank you, Peter. Thank you for inviting me, and greetings to all from India. Unlike Argentina, we are still in the process of, defining the regulations and the policies for genome editing in plants.
Dr.
So
Vibha, please take control of your presentation.
Yes. Thank you.
Can you please take the control? Click on the button.
Yes. Yes, I have taken. Thank you.
No, result you haven't taken the control. You can click in the upper side of Teams.
As I am clicking, the slides are moving.
But we can't see moving.
Can you now see?
No, you have to click take control in the upper side of Teams window. Take control. Okay. Okay. Now
It's done.
Now it's working. Please. Sorry.
Thank you. Thank you so much. Greetings to all from India, and thanks to the organizers for inviting me. As I said, unlike Argentina, India is still in the process of developing the regulatory policies for genome editing in plants. And I would just like to take you through this process and what is the emerging thinking and the drafts which are which look like the approach that is being taken. So there's a lot of excitement among the scientists from both public and private sector, and about the use of this novel technology for meeting different challenges in the agriculture. In Indian agriculture, there's a lot of research which has been initiated through the funding of the leading agencies, and our National Biotechnology Development Strategy is paying a lot of attention to this applications of gene editing in agriculture. There's a plan to have a mission program, and at the same time and other activities, infrastructure, etc. But focus also on formulation of the regulatory guidelines on gene editing. So as the discussions were initiated in the country about how to move forward, we looked at our existing regulatory framework for genetically modified organisms in India. So we have a kind of a rules 1989, which we call them, and these are the rules under which all the genetically modified organisms and products are regulated. As they were made way back in 1989, the rules are very broad in nature. Of course, the understanding at that time was much lesser as we have now, but the rules have not been changed. Instead, from time to time, different guidelines are issued to deal with the stages or different types of organisms. So we have this implementation mechanism where the Ministry of Environment, Forest and Climate Change, which is also the focal point for the Tait Protocol on Biosafety, which is the leading agencies, but it is supported by the Department of Biotechnology under our ministry to implement these rules. The rules are implemented through a kind of a series of committees, out of which the middle ones, the Genetic Engineering Appraisal Committee, what we call, is the apex body, and then we have different statutory bodies, the different institutional biosafety committees and RCGM. Like this, there's an elaborate structure which is there for genetically modified organisms. Why I shared this with you, because I'll be taking these names as I move along for the regulatory framework for gene editing. So the coverage of the rules 1989 in India, there is a one line which was added in our regulation, which said all the new gene technologies, in addition to cell hybridization and genetic engineering, will also be covered under the rules 1989. And when we looked at the definition of the gene technology, it meant that the application of gene tech, it seemed to be covering the gene editing as well. So this was a challenge, and based on this, as I said, there was a need for development of new guidelines and also seeking, some kind of an exemption if at all it is agreed upon. So as I mentioned, these rules are usually, dealt with by updating the guidelines from time to time. So the process was initiated by the Department of Biotechnology for the preparation of guidelines on genome editing. They prepared a document, but it covered, all organisms in one document, and it was opened for public comments in January 2020. However, different stakeholders, since it was open to the comments, one of the various stakeholders shared their opinion, but the key stakeholder for the topic that we are having today for agriculture, that was our National Academy on Agriculture Sciences. They took it very seriously, and they had a detailed, consultation with very, very important stakeholders. And what they said was that this is a technology that India needs, and this is a technology, all the issues like the questions being asked to Professor Dodna about off-target effects and all those things were discussed. And finally, the conclusions which came up was that there needs to be separate guidelines for genome edited plants because they have to be dealt with differently, and internationally accepted and clear definitions of SDN 1, 2 and 3 are to be included. So we have gone with the adoption of the definitions which were presented in the, which were sort of, presented in OECD and were more or less agreed by the participating countries. And then it was also discussed, recommended that genome editing is a precise and a targeted mutagenesis tool. So it should be exempted from the regulations and risk assessments under Rules 99, and there is a provision for such an exemption. So they also presented like a regulatory pathway where we had these definitions of the three, the three categories of gene editing in plants, and also, the pathway that was given that for SDN 1 and 2, we are only going to have a laboratory and the greenhouse studies. It will not go for, once it is said that it is free from the external DNA, then it would be treated in the same way as the plants of the, as the products of the traditional breeding. Whereas for SDN3, where we see, where there's a new insert, it would be treated in the same way as the genetically engineered plant. So this process, as Peter mentioned, that there is a committee which is working, and I've been part of that committee. After the review of the comments by the stakeholders, there was a stakeholder consultation and a new drafting committee was set up by the Department of Biotechnology, and I am also a member of that, and a draft guidance has already been prepared for in about 2 to 3 months work that was done. So now in the key points of the draft guidance is that SDN1 and SDN2 categories of genome editing technologies for plants that do not involve or carry exogenous genes, DNA, and are comparable to naturally occurring variants, these would be exempted from rules 7 to 11. But our approving authority is the Ministry of Environment, Forest and Climate Change. So they have been requested to grant such exemptions under the Rule 20 of the Rules 1989. However, recognizing that this technology can be, needs to be regulated at some point, the initial very early phase of the research and development, that is the prior to confirmation of the transgene-free lines, will be regulated, will be sort of, reviewed by our institutional biosafety committees, and for which we already have a guidelines for doing research activities in contained facility, which will be taken care of. So this draft guidance, which has been made by the committee, has already been approved by one of our leading, recombinant DNA advisory committee. It was presented by the chair of the committee to GAC, and the members were asked to give comments. It is in the final review by the GAC, that is the apex regulator in India. So this is where we stand, and I'll be happy to ask any questions. There's a lot of optimism, and subsequent to this, there has been some delay in, the final approval, and various stakeholders are actually, holding a lot of meetings and requesting the government to come out with this guidelines at the earliest, so that there is an enabling environment to take forward this technology in India. Thank you so much.
Thank you very much, Dr. Vibha. And if there's one question of clarification, we could take it. If not, please don't leave us, because we'll have a more general discussion in after the end of our final invited intervention. I can't see anything for the moment, so yes, please stay with us, and we'll see where we go with the discussion later. Now over to Australia. Thank you very much for joining us, Peter. I know that you have Carl there, but your name is Peter, so we won't go into why that is at this stage. But thanks very much. I know it's not a good hour for you, so let's give you the chance, and we take it away if we can have the presentation up.
While that's happening, I'll say thank you very much for the invitation to speak.
Please take the control of the PowerPoint. Okay.
Okay. All right, I'll dive right in. Brief overview. I'll give you a little bit of context. I'll talk about how the Australian regulation works. I'll talk about genome editing and what the issue is with regulation, and then I'll talk about our approach to regulation of genome editing as it currently sits. I like to use this slide as a segue to presentations. So this journey, I think, begins back in 1975 with the meeting of scientists at Asilomar when recombinant DNA was kicking off, and they took a very precautionary approach. And then the history what we're talking about is set by the history around 2000. Just get the laser pointer on. Most countries, and Martin referred to the Cartagena Protocol, so around 2000 we have GMO-specific laws and definitions. Then we move through new plant breeding technologies, which has morphed into being called genome editing, and as Jennifer spoke about, we have CRISPR. And perhaps to note, part of the issue is we have the European Court of Justice ruling. Okay, the concepts for recombinant DNA laws at about 2000 were that we had a new technology, that precautionary approach was still there, felt there was a need for pre-market assessments, but that traditional breeding technologies like mutagenesis might be excluded. So there are basically three approaches taken: GMO-specific laws, most countries in the world have done that; a novelty trigger, Canada and New Zealand are the two countries that I think are the preeminent examples of that, or to adapt existing laws without GMO-specific clauses. And I think the USA is the best example of that. So Australian laws for GMOs, GM food, and that relate to the discussion about genome editing. We have legal definitions sitting in the Gene Technology Act for GMOs and in the Australian-New Zealand Food Standards Code, with a process trigger, a definitional if gene technology was used, and then there's a science-based case-by-case assessment and approval pathway. I'll just show you briefly the definition for the food situation. Just you'll see that recombinant DNA features in that definition, and that the food is taken from an organism that was modified by gene technology. So I'm not from the food regulator, so I won't speak in any detail about their side of things. But I am from the GMO regulator, so we have a broad definition, an organism modified by gene technology with some exclusions, and we have a broad definition of gene technology, very broad, any technique, but with the keeping out of sexual reproduction, and also with the potential for techniques to be declared, and that's done in another piece of legislation called the regulations. In terms of those exclusions, there's a schedule for techniques that aren't gene technology. I won't read them all off. Basically, they exclude conventional mutagenesis techniques. And I'll come back to these other two schedules in a little while. Schedule 1, organisms that aren't GMOs or are not GMOs. Schedule 1B, quite a new one, specific definitions for organisms that are GMOs. And the regulations, as opposed to the higher level act, subject to fairly regular technical reviews. So the problem, back in about 2000, everyone had their different definitions, and your classical transgenic, those definitions would say that's a GMO. We fast forward to 2021, all of these new techniques, different definitions, and the question then arises, are those things still GMOs? So classically, 2000, you get the same regulatory outcome. In 2021, you've got different definitions that may result in different regulatory outcomes. And so, for example, you may have asymmetry where the GMO in country A is not considered a GMO in country B because of those definitions. So you've got uncertainty and even uncertainty in the wording of definitions, which was the case in our legislation. I'll keep moving along. In terms of the response to genome editing, I'll just say that in the food space, our colleagues have been looking at this and they have a review underway. I expect that you'll see something in this calendar year, but I won't dwell on that if you can look at their website for information. In the GMO realm of things, we did a review in 2017-18 and made some changes in 2019, which clarified the regulation around SDN1 and SDN2 in particular. There's also a policy review and its outcomes which are still ongoing. There was a reference in the review report to looking at definitions. Once again, I anticipate that you will see something about that in the coming months, but I won't say any more about that. That's a big P policy issue, if you like, not within the remit of the regulator. Too many fly-ins, sorry. Okay, so in this slide, if you concentrate on that dotted line, this is the outcome of the review and some of the rationale for it. The things that are red below the line are regulated as GMOs, and the things above the line, not GMOs. So you have the conventional techniques, and the review excluded SDN1 techniques where there was no template added. And so you have the SDN1 outcomes are very similar to those small nucleotide changes from conventional techniques. And if you look at that in terms of the regulatory wording, Schedule 1, and there are other items in these schedules, but for genome editing, organisms that are not GMOs, if it's an SDN1 modification and there was no nucleic acid template that was added to guide homology-directed repair. Mirroring that, also introduced in 2019, if oligo-directed mutagenesis was used, so you've got the introduction of some nucleic acid to direct the change, that is a GMO, and even if you use a site-directed nuclease, but you add a nucleic acid template to guide that repair to make the change, that is also captured as a GMO. I think I've maybe gone faster than I thought. One of the take-home messages I want to leave people with, particularly the scientists in the audience, is that it's the legal definitions that matter in the regulatory space. There are lots of terms of art about what genome editing is, what an SDN is, what SDN1 versus SDN2. And this is an example of a paper which incorrectly said that if no foreign nucleic acid was added, then it was not subject to regulation. However, as I've described, some genome edited crops are not regulated as GMOs in Australia, but some of them are. And so I'll stop there if anyone has any questions.
Thanks very much, Peter. And I know that you're one of the people that has additional material in your presentation, which we can share with others afterwards. Once again, if you could stick with us for the general discussion, but for the moment, any questions of clarification on the words of Peter? I nearly called you Carl because you. Sorry about that. Any questions of clarification? It doesn't seem to be the case at the moment. So, Peter, thank you so much.
Can I make one little comment?
Yeah.
Several people have spoken about the democratization of the technology, and Martin spoke about different multinationals versus small players. I've had the opportunity to be part of a number of other webinars over the last year, and a lot of those in the developing world, international agricultural research institutes, just to note that there are a really large number of small crop varieties versus the large commodities that the multinationals have done. So I think just want to emphasize that we are likely to see that application of this technology in a lot of species that we didn't previously see because they weren't big money spinners, but they may be very important locally. I'll stop there.
Thanks for emphasizing that. And I think it's one of the more important points that comes out of these types of discussions. So let me just make sure. I think now, oopla, I've got so much paper in front of me. So now we have Professor Masashi, and you're going to talk about genome editing in Japan. We've already heard Professor Downer mentioned the tomato, but would you like to talk to us? And thank you for staying within the time limit of, if possible, and I know we can share any additional information that you share with us with the participants afterwards. Thank you so much.
Thank you so much for inviting me. This kind of very important. So could you see, do you see my slide? Okay. No. Okay. So I like to talk about the current situation of the gene editing product in Japan. So first of all, I think all kinds of regulatory discussion in Japan started from this publication from Joint Research Center in 2011. So from this report, Japanese government started discussions. And after
Mr. Masashi, you have to take the control of the slides. It is a result that you didn't yet. Okay, now it's working, okay. Please.
So after 2011, the Japanese government started actively collecting information related to gene editing, and interest among scientists have grown rapidly. And that led to the establishment of the academic society, like Japanese Society of Gene Editing. Also, many government-funded research projects started thereafter, as you can see here. Now, today we have 2 gene-edited products which have successfully notified to the government. First product is high GABA tomato, mentioned by Professor Dauduna, developed by the University of Tsukuba. It was placed on the market from last month. The second notification was about madai fish, like we eat as a sashimi. So madai fish becomes bigger compared with the conventional one because of myostatin knockout. So from this slide, I would like to explain the regulatory issue of gene-edited products. First of all, I would like to explain general framework of GMO regulation in Japan. Basically, we are watching GM product safety from 3 aspects, from environment and food and feed, shown as here. So environment safety is covered by Japanese Carcinogen Act under the Ministry of Environment, MOE, and food safety covered by Food Sanitation Law under the Ministry of Health. And also feed safety is covered by Feed Safety Law by the Ministry of Agriculture. And based on existing GMO regulatory framework, Japanese government have discussed regulatory status of gene-edited products. Since there were various discussions within government section, it took time to reach conclusion. But the momentum to really tackle with this issue had heightened when the cabinet ordered each ministry to settle the issue by March 2019. So it's kind of top-down decision by the Cabinet Office. So in response to this order, and after the intense discussion, clarification was made by both Minister of Environment and Ministry of Health in 2019. I'm going to explain in detail in the following slide, but in essence, Ministry of Environment determined SDN1 not subject to regulation, but asked to provide information and made open to public. And the Ministry of Health also determined SDN1 and also in case of SDN2 on a case-by-case basis, not subject to regulation, but required prior notification and information made open to the public. So for the product intended to be released in the environment, MOE considered based on the definition in the Cartagena Act, which states LMO is organism that possess inserted nucleic acid processed out of cell. Basically, it looks the final product whether there is insertions of externally processed nucleic acid. And if that is absent in the final product, then it is considered to be out of the scope of the regulation. But even for those that are not within the scope of the regulation, if developers intended to place the product on the market, they are asked to provide information, and those in red circle are to be published on the Biotech Clearinghouse website. For food, Ministry of Health Council considered slightly different from that of Ministry of Environment. They asked whether the risk is similar to conventional breeding or GMO. Blue part is the conventional and red part is the GMO. Obviously, SDN1, a directed mutation without the use of template, considered to be similar to the conventional, and SDN3, as it contains foreign genes, is GMO. But for SDN2 in the middle part, they said that it's up to the Ministry of Health decision on a case-by-case basis. So all the developers has to undergo the prior consultation. Then Ministry of Health decide to whether the products fall within the scope of regulation or not. And if the product is out of the scope, not subject to the regulation, it will go on to notification process. But those products not subject to regulation must submit information, and the summary of the product will be posted on the Ministry's website. The information required are as shown here. So now I would like to touch upon the background of the Japanese Cartagena Act and show how the Ministry of Environment can draw the above-mentioned conclusion regarding gene-edited product. In order to understand the reasoning of Ministry of Environment, we need to understand how the Japanese government had stipulated the act based on the Cartagena Protocol. So this is very busy slide, sorry. This is a slide explaining the definition of LMOs in the Cartagena Protocol and the Japanese Cartagena Act. As you can see, the definition of LMO is almost the same, but there are slight differences in the wording. Japan maintains the word possess, but avoid using words such as novel or modern, because those words are very elusive and difficult to interpret. Instead, those words are replaced with the words such as nucleic acid and specified technologies. So as you can see, SDN1 are out of the scope because the organism does not possess the nucleic acid obtained through the use of recombinant DNA technologies. Also, SDN2 using templates and SDN3 are within the scope because they are prepared outside of the cell. So this slide explains about the policy for the Consumer Affairs Agency. The policy for labeling of gene-edited food was also issued by the Consumer Affairs Agency, in line with the distinction between made by the Ministry of Health. The Consumer Agency classified the gene-edited product into GM and notified product. They said the labeling is not mandatory. However, the agency also encourages the developers to provide information through labeling. So this is my conclusion. As you can see, maybe I like to skip this slide, so maybe you can see the summary of the Japanese situation. Thank you so much.
Well, thank you once again, Professor. And thanks for including a lot of additional information, which we will share with the participants through your presentation. So do once again, please stay with us if possible for the more general discussion. But are there any questions of clarification in the meantime? Michael Eckerstorfer.
Hello.
Michael?
Oh,
Hi. Yes.
hello. This is Michael Eckerstorfer. I'm working at the Austrian Environment Agency. Could you please, Professor, explain the criteria which are used to exclude one type of SDN2 application while retaining other types of SDN2 applications under the GMO law? Thank you.
Thank you, Professor. You got the question, I believe.
Thank you for your question. As far as I know, only the Ministry of Health maybe decides upon their decision whether the SDN2 using template can be regulated as GMO or not. So it's kind of based on the scientific decision, or like food safety, the concerns they found. Maybe in that case, maybe those products are under-regulated.
And you mentioned, I think, in your presentation, the important phrase case by case.
Yes, yeah. Case by case.
Yeah. Thanks, Michael. But once again, we will come back to the more general discussion later. Now we turn to, well, you're on travel, Dr. Olalekan. You are on travel and you're in Mombasa today. So thanks very much for joining us. And if I could just give a quick summary, you are from the African Biosafety Network of Expertise, which is run under the African Union Development Agency. Correct me if I'm wrong, but basically you're from an intergovernmental organization from Africa. So let's see if we can get your presentation running. Oh, great.
So please, you can take the control. Okay.
Okay. Thank you so much, John. Thank you, Peter, for the introduction. Yes, you are right. And I work for African Union Developmental Agency. Our head office is in South Africa, but our base in Burkina Faso. I came here for a meeting on capacity building for the Kenyan Biosafety Regulatory System Agency. I will quickly take us through the topic, the stature of genome editing. Oh, excellent, it's working. I need to give this perspective from African contents that most of the African countries, they base their regulatory system on Cartagena Protocol because most of the member states are signatory to this international protocol. And I will not want to define it again because Lema did a good job and other people that spoke before me also define it very well to save time. I felt I should give this audience to understand where African is coming from, where we are going, and showing the biosafety landscape in African countries. And if you can see, based on the Cartagena Protocol, they have moved, some of the country has moved non-adoption to the adoption of the technology. And we have a paper recently published this March that shows the interface between the regulatory approval and commercialization, which is the window that we try to fill in as a gap in that paper. Following this, there is a decision document from the head of state in Africa that have really shown interest that the genome editing should be harnessed safely.
We just lost his connection.
If somebody can help us help him reconnect, that would be great. Okay. Thank you.
Can you hear me now?
Yes.
Yeah.
Okay. So I'm trying to establish the source, the strength of the NEPAD bringing on board the use of emerging technology through the decision document by the head of states, which the member states have requested NEPAD to help in strengthening the capacity of the regulator. And this yielded a positive result during the last negotiation in Egypt, that all the member states were speaking with one voice because they have been trained for the essence of the technology, which has stopped the process of moratorium for that period, and which is still going in the same strength as we talk now. For the level, commended to the member states for taking the technology to use it, and those two technology are also requested to develop the guidelines on how this technology should be regulated. And the guideline, at least as I'm speaking to you, 2 countries have already made progress. But quickly to share with you the progress that have been made in some countries in Africa, especially Burkina Faso, that has taken the lead in the gene-edited rice, which they are presently working on at the low level, at the CFT level. And it's good to put to note at this point that member states, they have their sovereignty and they act independently. But African Union guide the process, give the decision document that the head of state have made as regard to technology and work in that process. But apart from Burkina Faso, we also have Kenya that have taken decision for the gene-edited product to allow at the research level at this point, because they are still working on their guidelines. This is just one of the pictures of the last visit we had in the Bobo Dioulasso. Bobo Dioulasso is a town in Burkina Faso where this research is ongoing. Now, for the case of Kenya that is taking the lead, they have the GM to be regulated, as Lema has also said, is a case-by-case base. And the flow charts that they have projected in their guidelines is what is what I'm projecting now. They have a flow chart that shows if it has a GM product, a GM insertion in it, will it be regulated? If not, will it not be regulated? So it is being treated or will be treated case by case. Similar thing is that case of Nigeria. I intentionally screenshot the Nigerian document or the guideline so that it can be a reference point for people that are interested to really download the guideline that have been developed. It's freely available on their website. It can be downloaded and they can read through and get the information in details. But it's good to quickly note here that this guide, Nigeria, they're just leapfrogging, because presently there's no scientist in Nigeria that is working on this product. But the regulatory body are already that any scientist that is interested in taking advantage of technology can go through the guideline and see the product is GMO, then it will be regulated. If it is not GMO, it should not be regulated. And I put all this paper also here that give the summary of the status of also here for us to have a reference in case somebody is interested. To come to a concluding point, it shows that African Union policies support the use of science, technology, and innovation for socioeconomic development of the continent. The emerging technology, which include gene editing, they have done a primary appraisal and they have given it a note to be used. And the member states have requested the African Union Development Agency, which where I work, to produce a guideline that could be a reference document for member states that is interested in the technology. I put this quote here for us to have a light that for us to meet the need of the moment, you cannot depend on the last technology of the old technology to get us to where the continent needs to be. I think at this point, I want to say thank you. If you want to get more information or you need personal things you want to get from us, our email is also put there so that we can get any follow-up. Thank you so much.
Thanks very much, Dr. Olalekan. I will say the same thing as I've said before. Are there any immediate questions of clarification? Otherwise, please stay with us and we will come back in the discussion session we will have. So, thank you. And thanks for persisting with us with the long distance technical arrangements. So now we turn a bit closer to home, as it were, and we go to the UK, and we will have a couple of interventions here. But let's start off with Louise Ball. You are from DEFRA, the Department for Environment, Food and Rural Affairs. And there's been a lot going on recently in the UK, so you will give us a quick summary, I think.
Yes, thanks ever so much for the invitation to speak, because it's great timing for us, because we've just published a government response to our consultation on the regulation of organisms produced by genetic technologies. So the consultation took place earlier this year. It had two main parts to it. I think the first part is the thing you're most interested in, and it's about changing the definition of GMO as it appears in English law. So I need to be clear here that it's a devolved issue. GM is a devolved issue in the UK. So in the different nations, we have different policies and regulations. So this is for England. And the language here is because of the language in our existing legislation, but it really aligns to the Cartagena Protocol. So when we're talking about organisms using genetic technologies would not be classed as GMOs if their genetic changes could have been produced by traditional methods. It's kind of aligned to the Cartagena Protocol. Could have been produced by traditional methods is aligning us to the sort of notion of new combinations of genetic material. But I'd also really much like to highlight that we are looking at reforming the whole of our GM regulatory framework. It's definitely something we need to consider, especially because our Prime Minister, in his inaugural speech when he took office, talked about liberating our bioscience sector from so-called anti-scientific GM rules. So that's very much a longer term ambition for us. So I'm not going to go through this slide because I don't have time. It sets out the results of the public consultation that we had. But I'd like to, I guess, make one point. We're very aware that most people don't really have an understanding of genetic technologies, gene editing. So it's something we're very much aware that we need to talk to people more about and get involved as much as possible in a public dialogue. And also we're going to be talking in much more detail to targeted stakeholders in the bottom paragraph. It sort of signals some of the issues that came up, which I suspect most people are familiar with. So finally, this is what our last paragraph effectively of our government response says, and it is talking again about this stepwise process that we're going to move through. We've got the two broad ambitions, first of all our step one, and then this longer term ambition in step two. So the first thing we'll be doing is bringing in secondary legislation to make field trials less burdensome. So there'll be a notification process rather than authorization, and no 9-figure grid reference, which 6-figure grid references, which is what we're requiring at the moment. And we're hoping to do that by the end of this year. The second step is, as I said, about changing the definition. So that's something we're very much looking into. It isn't entirely new territory for us. Again, that's why I guess we're starting with this. I think some of you will be aware that before the European Court of Justice ruled, we did have a couple of occasions where companies, organisations came to us for a view on whether we thought things were GM or not, and we said they weren't. So we'd already used our advisory committee and a scientific process to get to this point, and it's also aligned with our intervention to the EU Court of Justice ruling. So it's not entirely new territory for us, but we are doing a lot now. I think as the discussion has influenced, there's lots of issues and we need to make sure this lands properly and we do it properly. And the other areas that we need to look at, there are other measures, not just GM measures, that will affect whether gene edited products and the products of other technologies can get onto the market. So we very much need to look at those two. And then, as I said, the next big step will be to look at longer term reform. And I think Joyce is probably going to pick up on that more than me. So hopefully I've achieved that in 5 minutes, and I'll hand over to Joyce to talk her.
Thanks very much. I will take questions of clarification after Joyce Tait speaks, but Professor Joyce Tait from Edinburgh, and you were engaged in the report of the Regulatory Horizons Commission. So I hope you are with us.
Yes, I am. Yes, thank you.
Excellent news.
I don't see my slides yet.
Okay. Hoopla. Ah, great. Okay, we can see the slides.
Please, Dr. Joyce.
Okay, so I've clicked on take control.
Perfect.
And I should just mention that I'm a member of the Regulatory Horizons Council, and 3 members of that council were responsible for this report, myself, Matt Ridley and Andy Greenfield. And we're all got quite a lot of regulatory experience, some of us more in depth than others as far as genetic technologies are concerned. And we, if you look at the remit of the Regulatory Horizons Council, this is an independent expert committee supported by civil servants, and we're under the Department of Business, Energy and Industrial Strategy, so not directly responsive to DEFRA, but we've been having close discussions and interactions with DEFRA throughout this. We're part of a commitment from the UK government's white paper on regulation for the 4th Industrial Revolution to put the UK regulatory system as a whole onto a new, more innovation-friendly thing than it has been in the past, given that we're now no longer completely bound by the European regulatory approach. So the remit that we're talking about here, we were asked by the UK government to devise ways to regulate genetic technologies in agriculture in future so as to encourage safe and beneficial innovation. And it was related to our departure from the European Union, but also with the understanding that we do still have to trade with other nations in the world, and so we will have to have a regulatory system that is generally compliant with the standards that are in place elsewhere. But there is, you probably understand that the UK government has an ambition to become a leader in regulatory innovation, as well as just making individual regulatory decisions across the board. So this is part of an overall government agenda. And this slide is describing how we framed future genetic technologies for our study on the governance of innovative genetic technologies. And we conducted a background analysis that noted the lack of demonstrated hazards for the many products of straightforward genetic technologies that have been in place for 20 years. And beyond that, we have second generation genetic technologies such as genome editing, synthetic biology and engineering biology, which have the potential to transform agri-food systems, reduce greenhouse gas emissions, and contribute to climate resilience, sustainability, and biodiversity conservation. So we're talking here about the new genetic technologies, how they can usefully be regulated. And simple genome editing is one approach, but the more complex genome technologies like synthetic biology and engineering biology will also be extremely important in future. And need to be taken into account. So we took a relatively open remit and decided to look beyond simple gene editing, SND1 and SND2, and look at what would be needed to give all genetic technologies a more science-based and pro-innovation route to market in other countries, focusing particularly on a product-based approach and what it might look like. So this is a longer-term objective of our study, which was to look at how we could propose a regulatory system that would work, that would be product-based, with minimum reliance on arguments about processes and what processes were regulated by which instrument and so on. So it's relevant to gene editing, and what we're saying is we must be careful that while we're doing the current regulatory adaptation to meet the needs of simple gene editing technologies, we don't actually introduce something into the regulatory systems which will make it much more difficult in future to regulate these other much more powerful genetic techniques like synthetic biology that involve trans-species genetic transfer. So our proposed regulatory approach, that there's a link to the website for the report itself on the first slide, and you will be able to read this if you take a look at the report on the web. But our proposal is process-based for the very first regulatory decision, which is looking at whether the genetic modification that has taken place, if something comes under the definition of the Cartagena Protocol definition of an LMO, then it will be looked at by the regulator initially to decide whether there is any grounds, scientific grounds, to expect any risks arising from that process in the product. And beyond that point, it's regulated by what we call the sectoral regulatory system for the kind of product that it is. So if it's a novel food or a novel animal feed, it goes straight to the regulatory system for that type of product. And there is no additional consideration if there are no risks identified. There's no additional consideration of its role as a genetically modified or gene edited or synthetic biology based organism. So our proposal is slightly different. I think we tried to be as inventive and innovative as we could. We took account of all the other regulatory systems that we were studying throughout the world. We tried to do something that went beyond what most other people are doing. I hope we've come up with something that is that takes things forward in a productive and helpful way, but also that allows the UK to develop its current approach to gene editing as is being proposed by DEFRA, and allowing us to make quick wins in that area following what's happening in the rest of the world. So I'd be very interested to discuss this with anybody either now or be outside this meeting if anybody is interested to look at our proposals and discuss them with us and help advise us on what needs to be done in future in this area. Thank you very much.
Well, thanks very much, Joyce. Yeah.